Every single living thing is built from a blueprint, a genetic code that dictates every single detail about us. So today, we're going to take a look at that blueprint and explore what happens when the instructions have an error, one that gets passed down through generations. Okay, let's really dive into this. We think of our genes as this amazing code that makes us who we are, right? But sometimes that very same code can be the source of disease. So how do we even begin to wrap our heads around these inherited conditions? Well, it all starts with knowing exactly what kind of error we're looking for. To get a real handle on these diseases, we've got to classify them first. Think of it like this. You're in a massive library. You need to know if you're looking for a single misspelled word in one book or if you're trying to find a whole missing chapter. They're two very different problems. And when you boil it down, genetic disorders really fall into two big buckets, and the scale of the impact is totally different. On one side, you've got Mendelian disorders. These are like a single critical typo in the genetic code. But on the other side, we have chromosomal disorders, where the error is so much bigger. It's like an entire page of the instruction manual is duplicated, or maybe missing altogether. So let's start small. Let's start with that single typo. Mendelian disorders are caused by a mutation in just one gene. And what's really interesting is that they follow these predictable patterns of inheritance, like dominant, recessive, or X-linked. For today, we're going to focus on a classic case to see exactly how this works. What this really means is that we can put on our detective hats. By studying a family's history, we can often trace the exact path of a single faulty gene as it gets passed down from one generation to the next. Okay, so to really see just how much chaos one tiny single gene error can cause, sickle cell anemia is the perfect example. It's a disease that messes with hemoglobin, which is the super important protein in our red blood cells that carries oxygen all over our body. Now, sickle cell anemia is what's called an autosome-linked recessive trait. That phrase tells you two really important things. First, the gene is on a non-sex chromosome, an autosome. And second, recessive means you have to inherit two copies of the mutated gene, one from each parent, to actually have the disease. If you only get one copy, you're a carrier, which means you don't have the disease, but you can still pass that gene on to your kids. And this is the absolutely mind-blowing part. The entire reason for this devastating disease, it all comes down to a single point mutation. A change in just one letter of the genetic code in the gene that builds hemoglobin. So let's break this down. It is an incredibly tiny error. In a normal healthy gene, the DNA sequence GAG tells the cell to put in an amino acid called glutamic acid. But in sickle cell, just one letter flips, changing the sequence to GUG. That tiny change now codes for a totally different amino acid, valine. And that's it. That one little swap is the entire root cause of the disease. And this is where you see the domino effect. That one tiny amino acid change completely alters how the hemoglobin protein behaves. When oxygen levels are low, the mutant hemoglobin molecules literally start clumping together. They polymerize. This forces the red blood cell, which should be a nice, flexible disc, into this rigid crescent or sickle-like shape. And those sickled cells can get stuck, block blood flow, and cause all the terrible symptoms of the disease. So we've just seen the incredible impact of a single typo. Now, let's zoom all the way out. What happens when the error isn't just one letter, but a whole page of the blueprint is missing or duplicated? Yeah, so we're not talking about a single gene anymore. A chromosomal disorder is a problem with the structures that carry thousands of genes. It's really a numbers game. Either you have too many chromosomes or too few. So for us humans, the standard balanced blueprint calls for exactly 46 chromosomes. They're neatly organized into 23 pairs inside pretty much every cell in our body. So a perfect example of this is a condition many of us have heard of, Down syndrome. What exactly is different in the genetic blueprint of someone with this condition? Well, instead of 46 chromosomes, their cells have 47. This usually happens because of a little mistake during cell division, where the chromosomes don't separate properly. This can lead to an egg or a sperm cell ending up with an extra copy of a chromosome. And that extra chromosome, it's specifically an extra copy of chromosome 21. So instead of the usual pair, individuals with Down syndrome have 3 copies. And that's why you'll also hear the condition called trisomy of 21, tri for 3. And you can really see the massive, widespread impact of a chromosomal error here. Because an entire chromosome is duplicated, carrying hundreds of extra genes, the effects are seen all over the body.
It leads to a very distinct set of physical characteristics and also developmental delays. It's just a completely different ballgame from the more targeted effect of a single gene mutation.
Okay, so we've got our two main types of errors, the typo and the missing page. But how do scientists and genetic counselors actually track these things through a family's history to figure out risks and inheritance patterns? Well, they have this amazing tool called pedigree analysis, and this is really the detective work of genetics. By mapping out a family tree and carefully noting who has a particular trait or disease, geneticists can actually figure out its pattern of inheritance over many generations. Now, it might look a little complicated at first, but there's a standard set of symbols everyone uses. A square is a male, a circle is a female. If the symbol is shaded in, it means that person is affected by the trait we're studying. And the lines, well, they connect parents to their children. It's basically a visual language for genetics.
Okay, so let's put our detective hats on again. Take a look at this pedigree chart. You see two parents in the second generation who are totally unaffected by a condition, but they have a child who is affected. Remember what we talked about with sickle cell anemia? When you see this pattern, where a trait seems to skip a generation, what does that immediately tell you? What's going on here?
If you said autosomal recessive, you are spot on. That pattern is the classic signature of an autosomal recessive trait. It means that both of those parents have to be heterozygous carriers. They each carry one copy of that recessive allele, but because it's recessive, they don't show the trait themselves. This is the incredible power of pedigree analysis. It reveals the hidden genetic story in a family and helps us predict future risks. And look, at the end of the day, this isn't just some academic exercise. Knowing the difference between a Mendelian typo and a chromosomal missing page is absolutely fundamental. It has huge, real-world implications, and it guides everything from making a diagnosis to providing genetic counseling for families. So, whether it's a single letter change or a whole extra chromosome, the story of genetic disorders is written right there in our DNA. And as we get better and better at reading that story, we're not just learning how to diagnose diseases, we're starting to learn how to rewrite the future of health itself.